my name is Dr. Christopher Ahern, and I'm going to give a talk today about some work that's come out of my lab at the Carver College of Medicine at the University of Iowa in Iowa City, Iowa. Today's work was a collaboration between my lab in the Department of Molecular Physiology and Biophysics and that of Ashlyn Williams in Psychology in the Iowa Neuroscience Institute, as well as a postdoctoral fellow in her lab, Karina Kruth, and a postbac in my lab, Tierney Grisolano. And so what I want to talk about today are premature termination codons as a cause of human disease, and how they arise from point mutations from the genetic code, giving kind of everyone a basis for premature termination codons. I'm going to be abbreviating that terminology PTC. And a method that's emerged from my research program that uses small molecules called transfer RNAs as a potential way to repair or correct these PTC disease-causing codons. I'm going to talk to you a little bit also about a screen that we ran involving different types of transfer RNAs and how we have identified multiple unique transfer RNAs with high potency and fidelity for correcting premature termination codons in multiple diseases, including SCN2A. And then I want to talk a little bit about some of the challenges and next steps. But before I get into the data and the introduction, I'd just like to give a big thanks to Leah and everyone at the SCN2A Foundation. It's always inspirational to work with you guys, and I'm really excited for this presentation today to get your feedback on our work so far and really to work with you going forward. So premature termination codons are a major cause of genetic human disease. There are almost 5,000 estimated rare diseases in the world, and of these, 80% are genetic in basis, which means they come from alterations in the genetic code. When we look at all these genetic diseases, 10 to 40% of any one of them are caused by nonsense mutations, which we also call premature termination codons. And what that means is that's a mutation that arises within the genetic code which causes the gene to be stopped read in its tracks at this premature termination codon. And because it's a composite of so many different diseases, they're very rare individually on their own. There's many rare causes of premature termination codon disease, but because it's such a broadly impacted population, you may be rare, but you are not alone. And in the group and in the mass, we hope that we can find a cure for these PTC diseases. And along that same thinking, if there's a common cause for all these different types of human diseases caused by singular PTCs, is there a common cure, or is there a common method that could be developed or used to repair or correct these PTCs? And in particular, large genes which contain PTCs are more likely to have protein terminations occur in them just because of the large size of the genes. But because of traditional gene therapy, for instance, gene replacement, large genes are very challenging, such as ion channels like SCN2A, simply because they exceed the payload of most of the AAV delivery methods. Furthermore, because many PTCs are unique throughout a gene, the preciseness that we've grown to love with CRISPR and base editing approaches will be difficult to apply to all the different PTCs because they all come in different contexts. So I want to talk to you today about this new method that may be used or may be developed eventually to repair PTCs generally. So what are PTCs and where do they come from? So the genetic code is made up of essentially 4 letters, A, T, G, C. And this genetic code gives rise to the variability of all the biology on our planet. And so if you have a string here of nucleic acids that I've strung together, what the first thing is the genetic code is based on is these are the letters of life. And the first thing that needs to happen is they need to be broken up into individual words. And so if we break these into individual words, each word is made up of 3 of these codons. We call it a triplet codon because any 3 of these nucleic acids come together makes its own letter or part of a word. And the periods at the end of these sentences or words are formed by what are known as stop codons. Whereas many triplet codons can be indicated to be an amino acid and then form part of a protein, stop codons, there are 3 different types. And that's TGA, TAA, and TAG. Those are the different nucleic acids that make up the individual codons. And so whenever you come across this stop codon, that stops the word in its tracks, and then that's the end of the word or the end of the sentence of the protein. So now if we were to decode this, we know that this encodes for a C, and this encodes for a U, and an R and an E, and then we have SCN2A. So each one of these triplet codons has meaning, and that how this triplet codon gets turned into a letter, and then those letters turn into sentences in the form of proteins. In the form of SCN2A, where over 1,500 different letters come together to form an incredible machine called a voltage-gated ion channel. So if this is one way we think about it, the stop codon comes at the end of the sentence, as it's supposed to be. But what happens in premature termination codons, if you simply have one letter that's mutated to the wrong nucleotide, in this case we have a spurious T showing up in the middle of our sentence, what happens is in the middle of our sentence, cure SCN2A, we run into a stop codon, which halts the progression of that sentence middle of its tracks. And so what we wanted to do is there a way, using transfer RNAs, and I'll tell you about those guys in a second, how could we reintroduce this reading frame so we could finish that sentence? And so when we back up just for a second, let's think about the complexity of biology and the genetic code, as we call it. And so what you're looking at here is a graph of all the different codons that make up the genetic code on planet Earth and all of biology. And so what we have here is each one of these triplets, as they're shown here, these three letters together, each one of them gives rise to a different type of letter or word, which is shown here. And these are the amino acids. These are the bases, and these encode for the amino acids. As I said before, there are three stops that are built into this language, and those encode the end of a word or the end of a sentence. However, if you look closely at this genetic code, what you'll discover is that there are 10 different types of amino acid codons which are uniquely vulnerable to conversion to a stop codon. Said another way, a simple single mutation in any one of these additional red boxes here of these codons will convert them to one of the three known stop codons. So there are 10 types of amino acids, which is a broad swath of all of biology, which can accidentally be converted from a go codon or a sense codon into a stop codon. And the way that these triplets get actually kind of translated, as it were, into the amino acids is through these transfer RNAs. These are short 70 base pair nucleic acid fragments that really decode RNA into protein. They're the ultimate translation device. And so what we wanted to do was to take the idea of a transfer RNA with the given premise that transfer RNAs, what they do for a living is essentially decode nucleic acid triplets into amino acid letters. Could we get them, these tRNAs, these transfer RNAs, could we somehow engineer them so that they now were able to decode stop codons and repair them back to their natural amino acid? So in this schematic here, these arrows are essentially what we would think of as transfer RNAs. For every codon, there's a novel type of transfer RNA, and this is actually a picture, an actual atomic resolution structure of a transfer RNA. You can see this compact little structure, and the way that this molecule works is that this region right here, we call that the anticodon. And what that refers to is that this part is the back end of the arrow that sticks right to this nucleic acid triplet, and it senses that triplet and it fits perfectly to it. And what it does is it delivers then the amino acid that goes with that triplet and designs it so it builds it right into the sentence, right where it's supposed to be. So these are the transfer RNAs. You can think of those as the arrows in this diagram that decode the nucleic acids to the amino acids. And so what we wanted to do was to build a system where we change that stop into a go, essentially changing the nonsense or the premature termination codon back into a sense codon, rebuilding the rest of the SCN2A channel. And in order to do that, what we wanted to do was to form a library of transfer RNAs. Given the human genome is known, we know that all the genes of the transfer RNAs are available to us scientifically. And so what we did is we systematically perturbed this anticodon region such that it no longer had the ability to bind and interact preferably with its sense codon here or here or here, but instead preferably would bind to this type of codon, a stop codon, and bring it back to life, and in doing so encode the right amino acid at that site. And so the premise of the screen that we ran is that there are essentially 450 different human tRNA genes. So it's a large category of genes to work with. And what we did was we systematically changed this anticodon region to the point where it would no longer interact with what we call its cognate codon, the triplet that encodes for an amino acid, but instead we looked for emergent behavior in the form of tRNAs that would instead interact with stop codons in the middle of genes and reform new peptide bonds such that the stop codon no longer read for a stop, but instead could be changed back into a sense codon, and the amino acid would be inserted at that site, more or less repairing or correcting that stop codon lesion in the gene. And so in order to do this, we set up a large screen that looked at all these different tRNAs just to look objectively which ones may work as stop codon suppressors. And so we performed what's called here a high throughput screen, which is unbiased and allows us to just let the data tell us what it sees in terms of which tRNAs are open to this manipulation and have this behavior. So we based this screen on a reporter in which we put a stop codon into. This is luciferase. This is a version of the enzyme that lights up fireflies in the summer around this time all throughout North America. And so this enzyme forms a bright substrate which we can see through fluorescence. So we can measure the activity of this enzyme by just looking at it with sophisticated instrumentation in the lab that will measure the light output of this little enzyme. And what we did was we engineered a stop codon into this loop right here such that it broke the enzyme and it no longer fluoresced. And then we put this in with our library of transfer RNAs, and we looked to see which one of these transfer RNAs have the ability to repair the luciferase and turn the light essentially back on in this enzyme. And so this is the full output of the library screen that we ran. And what you're looking at here is by color are the different gene families of transfer RNAs. So the first thing you'll notice is that for each color, so for instance, this is glycine, and all these glycine tRNAs have been converted to UGA suppressors, which is a stop codon. And then what we're looking here on this axis down here is the ability of it to rescue that luciferase gene. So for instance, in any given family, this red line right here, that means it has no activity over background. So those are examples of transfer RNAs which were intolerant of the ability to kind of turn that mutation in the anticodon into a new functional suppressor tRNA. However, what you can see also is that, so for instance, arginine is one of the most common stop codon types. Here's a stop codon for arginine, converting UGAs back into arginine. And what we see here is these bars here represent between 100 and 1,000-fold over background, which means that tRNAs, these individual tRNA sequences, have the ability to convert a stop codon in that luciferase to a functional luciferase at a very high degree. And so what these individual colors represent are every known point mutation that causes a PTC to cause human disease. Said another way, this library covers every known human PTC that causes disease. And so now what we want to do is to understand how well do these work in different contexts? Do they encode the right amino acid? What about real stop codons that end sentences normally in a biological environment? And can we pave a way to a more clinical kind of outcome of using this library to find a way to basically cure every known PTC, human disease, with a primary focus in my lab really being on SCN2A? And so one of the things is we always say is seeing is believing. So this is just one example of many of the different types of tRNAs that we use. So we often use GFP as well, which is a fluorescent molecule from a jellyfish in the ocean. So it's green fluorescent protein, and we can express this green fluorescent protein in a cell line in the lab, and that's what we've done here on the left. And so you can see each one of these green specks is a cell that's expressing GFP, and they light up in a fluorescent way. If we throw in a stop codon in the middle of that GFP gene, we essentially shut off the gene. It no longer can make GFP, and it's non-fluorescent. However, if we co-express this GFP TGA mutated GFP simply with a couple pairs of that arginine UGA suppressor, what you can see is it more or less completely revives that GFP signal to be near more or less the control or full-length GFP. So this gives you an example of just seeing with your own eyes how potent can these tRNAs be in terms of turning a stop codon back into a codon that's read to form a normal enzyme, or in this case it's a fluorophore GFP. And so one of the first things we wanted to do was to look at, okay, if we can mess with the anticodon, can these tRNAs still put in the right amino acid? Can they put in their cognate amino acid, or do they have high fidelity in the way that they rescue? And so we turned to a technique called mass spectrometry. And what that allows us to do is to look at the site of incorporation and measure the mass, the actual weight of the protein, and know which amino acid is there based on the weight of the protein. And what you're looking at here is raw data from mass spectrometry, and it's hard to describe in simple terms, but what these are all the individual masses for this fragment. And what it's telling you is at this site, we have an asparagine that's supposed to be an asparagine, and that's what we've done here. We have an asparagine, and we've mutated it to glycine with our tRNA, and that's exactly the mass that we see there. And also we have an asparagine, and we changed it into tryptophan, and again, we have the exact mass. And so what this preliminary data tells us is that these tRNAs are also encoding the right amino acid, which, as many of you know, for SCN2A is incredibly important because SCN2A is very poorly tolerant of missense mutations. So it's important that we're getting the right amino acid encoded back into the ion channel. If we look at different types of stop codons, this is a glycine tRNA in this NEST experiment, and we've looked to see can it suppress really the one that we're directing it towards. In this example, it's UGA, or does it hit the other ones? And what the data clearly show is that the luminescence, in this case it's the nanoluciferase again, is only restored when you have the right stop codon in place. And then lastly, we looked at common quote-unquote read-through drugs. These are small molecules based on aminoglycosides, which essentially cause the ribosome to skip and shutter and go over stop codons. We wanted to compare our tRNA approach to these small molecules. And what you can see here is gentamicin and G418. Those are traditional kind of small molecule aminoglycosides, read-through drugs. And what you see is that the tRNA vastly outperforms the read-through drugs as well. So we've established we have a large library of tRNAs that covers all the human PTCs. They seem to be putting in the right amino acid. They're highly selective for the right stop codon, and they seem to be also performing better in simple cells anyway compared to small molecules. And so then the next really big question was to assess, do they interact with real stop codons that end protein translation generally? So every gene that encodes for a protein has its own reading frame, and it ends that sentence or the last word of that translation, as it's called, with a real endogenous stop codon. And one of the things we wanted to look at is it possible that the tRNAs that we've identified in our screen, do they only hit the premature termination codons, or is it possible that they could hit the real stop codons, which would be a very serious off-target side effect? And so we turned to a method called ribosomal profiling. And what ribosomal profiling is it looks at these machines called ribosomes. And so what ribosomes do is they cruise across RNA in the cell, and what they do is they run down the length of the RNA and they make protein as they do so. They're essentially the catalytic center or the factory for translation of turning RNA into protein. And at any given time in a cell, you'll find RNA sitting all over, or you'll find ribosomes, I should say, found at all different sites all over any given RNA transcript. So what we can do is we can take all the RNA out of a cell that's expressing one of these tRNAs, and we can look to see where are these ribosomes, with the main question being, is it possible that some of these ribosomes are showing up in the region after the stop codon? We call that region the 3' UTR. At the beginning of the gene, it's the 5'. At the end of the gene, it's the 3' based on the chemistry of the nucleic acids. And this stands for the untranslated region, which means that essentially, in a traditional sense, you will have a stop codon, and that after that stop codon endogenously, it will be untranslated because the protein translation has stopped. And so what we looked at is where are ribosomes sitting on those RNAs in the entire cell. So this is the whole exome ribosome experiment. And what we've done is we've looked at different cell types that are expressing some of our highest performing or best suppressor tRNA in individual experiments. And what you see is data that looks like this. So what you're looking at this axis is the reads, which is how many times do you find a ribosome at that site. And this is at the specific site of every known RNA that's lined up in this analysis such that the stop codons are all kind of held together, and then we back up and say, where are all the ribosomes at any given time in that cell? Okay, so hopefully if you made it with me through that explanation, this data is actually quite easy then to see or to understand. And so what you can see is there's this sawtooth pattern. And what that sawtooth is actually those are ribosomes that are hopping down the RNA and they're reading individual codons. Remember in the beginning I talked about how the codons are read as amino acids? So these codons, in this instance, you have the ribosome moving into position, a tRNA coming in and delivering an amino acid. That's forming another peptide bond, and then it moves down and down and down the RNA until it gets to the very end of the reading frame and it bumps into the real stop codon. And then you see this large density here where all the ribosomes are kind of bunched up at the end of the RNA. And what's important here is that in the presence of any of our transfer RNAs that are suppressing stop codons, there's no evidence that you're reading through those stop codons in a biological environment. This was a surprising discovery. It is fortuitous for the use of these transfer RNAs as potential therapeutics or RNA correctors in a clinical sense, but it's also something that we're very interested in. It's meriting a lot more investigation on our part, but what we can tell you right now is that it does not appear that these tRNAs are promoting read-through of these quote-unquote real stop codons. And so the last thing I want to share with you is some data that we've captured in the last year looking at specific PTCs in SCN2A. And so what we've done is we've made these premature termination codons in the SCN2A gene and expressed it in simple cells, hex cells, and then we electrophysiologically assess the density or the amount and the behavior of the channels that are expressed on the cell surface. If you're used to seeing SCN2A, you may be more accustomed to these waveforms. And so what we're looking at here on the left is the electrophysiological recording, as it were, of the SCN2A channels that are expressed on the cell surface. The SCN2A protein is shown here. It has 24 transmembrane segments. Each individual kind of domain, we have 4 domains within those 24 segments. Each individual domain can be broken into a voltage sensing domain and a pore domain, and this thing folds up in an exquisite manner to form what's called a voltage-gated sodium channel. And so when we look at the activity of voltage-gated sodium channels electrophysiologically, what we're doing here is we're giving a series of depolarizations. We're essentially changing the voltage in the cell. And when you change the voltage in a cell, sodium channels turn on. That's their main role to drive the upstroke of the action potential neurologically. And so they're very sensitive to changes in voltage. And what you're looking at here is the activity of maybe 1,000 or 2,000 sodium channels that are expressed in this particular cell. So this is representative of a wild type or a normal sodium channel. So the first thing we wanted to do was look at arginine because it's a common PTC, and so we chose 2 different arginine mutations: R607TGA, which is near the N terminus, the beginning of the gene. And then we looked at 1626TGA near the very end of the gene. With the simple question is, does the context of the PTC and the surrounding sequence, which is very different for these 2 sites, does it matter? Does it affect the ability of the tRNAs to read through or to correct that stop? And as you can see, both of these recordings, when we put them in the presence of an arginine suppressor for that stop site, we more or less recapitulate what is near wild type levels of channel activity. For both of these sites, in the absence of a tRNA, there's no activity whatsoever. It's essentially as if the cell is quiescent and it's not expressing any channels. So this large activity, what we're seeing is due purely to the tRNA rescuing those PTCs at those 2 sites. And so it does not appear from what we've done so far where the PTC is. It can rescue well in the beginning and it can rescue well in the end of the gene. So then we quickly looked at a number of other different types of tRNAs and locations in the gene, as well as different stop codon types. And so we wanted to look at a serine mutation, so we pulled a serine tRNA out of our library that I showed you a few slides back. And we took this serine tRNA and we co-expressed it with this stop codon, and we get decent rescue there. Similar with this glycine, again, pulled one of the better glycines out of our library, and again, we're getting good rescue at that point. And then tryptophan, this is even a different type of stop codon. So the other stop codons were TGAs. This one's a TAG, an amber stop codon. And here we get very nice rescue across the board. So this is a data set that is living. It's pre-published. We're working on it still. But what it appears, at least the early data is showing us, is that it doesn't matter if the site doesn't matter the type of amino acid, doesn't matter even the stop codon, there's a tRNA in the library that can be used to repair it. And SCN2A seems uniquely susceptible to this type of correction. Now again, because this is only for stop codons, it's not going to work with missense mutations, at least not the current iterations that we've developed in the methodology, nor will it work in frame sense mutations, again, not with the current iteration of the methodology. I think when you start working with something like transfer RNAs, anything is possible down the road, but at this stage, we don't think we have any solutions for those other types of mutations at this point. And so in summary, what I hopefully have been able to show you today is that we have multiple tRNAs for every known type of stop codon causing PTC, including every type. These tRNAs have limited interactions with protein with real stops. That's the ribosomal protein assay data I showed you. They have high fidelity encoding, and the repair is robust. We're seeing very good repair efficacy in the 40 to 80% range, which is better than any of the aminoglycosides that are currently known, and we're quite happy with that rescue level. So now we have some next steps. We have a lot of challenges. The first, we need to test more tRNA types. I showed you maybe a handful right now. We need to get better coverage across the board of different tRNA types and different stop codons. And do these tRNAs, the next thing we want to understand is, do they antagonize a process called nonsense mediated decay? And this is a biological process by which RNAs that have PTCs in them are degraded. And so if we have a degradation of our RNA, we're going to lose the ability to rescue those channels. So if we can express these channels early enough, or these tRNAs early enough, then we can hopefully kind of compete out that NMD process and make more channels. We're also working with a number of groups, Children's Worldwide, as well as LEA and the SCN2A Foundation, to look at human iPSC neuronal cultures of SCN2A PTCs, and that's a lot of work that Dr. Williams is doing with my group. And so human iPSCs getting delivery worked out in the form of AAVs of these tRNAs into iPSCs is a really big step for us preclinically, because rescuing something in a hex cell is really quite straightforward. Rescuing it in a human iPSC is another story that has been derivatized to become a neuronal cell type. And so tRNA delivery by viral AAV is a big issue for us. And you know, the early signs appear to show that we can actually get decent delivery. So we're pretty excited about that. The nice thing about transfer RNAs that I didn't get a chance to necessarily dwell on too much is that transfer RNAs are very small. So normally the AAV payload might be 4 or 5 kb. Transfer RNAs themselves are only 100 base pairs. So you can imagine a scenario where you could put in many different types of tRNAs within a single AAV to deliver tRNAs to different types of PTCs. And lastly, in terms of how do we get from here in the academic setting to out there in the clinical setting, and the only really viable way to do that is through partnerships, working with other groups, with the SCN2A Foundation, and with biotechs to help kind of cover that valley of death that all new technologies have to get across. And the first steps of that have been completed in that we have IP that has been secured, and we're very close to securing some biotech partnerships right now that will really accelerate this therapeutic pipeline towards the clinical setting. And so with that, I'd like to thank you for your attention for my talk. I will look forward to learning more in the SCN2A Foundation annual meeting. And I'd like to, this quickly before I go, thank everybody in the lab. Of course, there's the current lab members that contribute to an amazing environment of scientific serendipity and discovery at our lab at the University of Iowa, and also as well as our SCN2A collaborators, Ashley Williams and Dr. Karina Kruth, who are a fantastic team of scientists to work with. Our funding agencies, NIMGS, which is a basic funding agency at NIH, but it's through that basic research that we're able to discover this novel ability of transfer RNA. So basic research really does matter, and that it helps provide the environment for discovery towards clinical settings. And then lastly, this work was directly funded by Safari Initiative by the Simons Foundation. And with that, I'd like to thank you very much.
Great, thank you, Chris, for your presentation. It was pretty amazing, all the work that you've done and continue to do. And we have a couple questions here. So you briefly mentioned that, you mentioned that this technique works only for nonsense variants and frame sense, correct?
Yeah, that's correct. The current iteration is that it's directed. It was designed to go after the premature termination codons. That's just one variant, as we know, of all the different type of genetic lesions that can occur in SCN2A in any gene. It covers a big chunk, 10 to 40% depending on the gene family. So we're happy with that, but we're not content. And I think that because we've learned that transfer RNAs are quite plastic in their ability to be re-engineered, we now are kind of looking at them with fresh eyes and wondering if there are ways that we could engineer them to take care of frame shift mutations, which are almost just as common, and they can be found in almost any genetic form. So our eyes are on those downstream developments, but that would be a new type of methodology at this stage.
How many variants have you studied? SCN2A variants that have this nonsense?
Those that I had on there, I think there were 6 that we've done so far. And since I put that slide together, we've done 2 additional. We have a C959, which is also one of our iPSC lines, and then we recently just started going after a tyrosine 84, which is also one of our iPSC lines, with the idea of trying to really build a vertically integrated approach, as we would say scientifically, that if we can take something in a very simple environment like a transiently transfected cell and deliver a correction at that stage, can we then step it to iPSCs, and then can we step it then to an animal model, and then all the way up to where we would be towards the clinic, at each stage kind of validating the technology. So we have a good sense. We've been trying to cover as many different types of stop codons and within the size of the gene, the positioning, as well as the different amino acid types. So we've just been kind of fanning out and building tools as we go.
Great. What's the anticipated timeline, do you think, that we will, to get to clinical trials?
That's tough, and I think that any academic scientist would really struggle with that. What I can say is that my understanding is that if all goes well, we could be in the 3-year to 5-year range, and we would hopefully like to accelerate that. Partners with biotech and additional resources will accelerate those timelines, because what we'd like to do is try and find a way to know how far can we get as quickly as possible. We like to say we want to fail fast in the lab, because then we can find ways to fix things and correct them. And so currently, we're on track, but we want to get through the iPSCs, animal models. So I would say in the 3-year range we could be there. But again, that's pending so many things when you have a new technology like this. But it's just one of many things that, as you've seen in this meeting already, there are so many different approaches that are coming out, and all of them together, I think, will come together with different types of cures. If you look at the CF Foundation, for instance, they've had a really systematic approach to going after the different types of mutations that affect the CFTR ion channel. And they've been successful at systematically taking off, piece after piece of that pie chart of all the different types of mutations. And I think if you look at the types of technologies that are being developed for SCN2A, we could hope for a similar outcome, and that we can hopefully be there in a few years.
Great. Now, what is something that families can do? What's the call to action, or how are you going about recruiting or needing participation?
Well, at this stage, we're going through the channels that are provided through us with the SN2A Foundation to secure iPSC lines. And absolutely, if there's other lines that could be made available, I think those will always be of use. So that's something that people can do, is work with their physicians in their local centers of excellence to provide iPSCs that can be used then as research reagents. And that wouldn't be just for this research program in my lab, but those would be reagents that would be very valuable across the board for many different types of technologies that are being developed.
Great. Now, does the age of the patient make a difference in treatment options for future?
I would say at this point, we don't know enough yet. Although what we've seen is that some of the work of Dr. Bender from UCSF, from my understanding, there is some hopeful data out there that shows that SCN2A restoration in a developed animal restores some of the synaptic function. So there is a possibility that restoration later in life could lead to more neurocognitive restoration. So I think that there's hope there for not just SCN2A, but many different types of genes. It appears that restoration later on does bring back a lot of the function. So I think we're hopeful for that possibility, but I think until we get that far, we don't quite know yet. But there's reason to have hope that there could be restoration.
Definitely. Now, with your methodology, would the plan be to do like a one-time like infusion of some sort, or would it require multiple?
At this stage, we're really hoping to work with the delivery such that it'd be a one-time or a rare dosing event. And so a lot of that comes down to the AAV types and how it would be delivered. But you know, on our side are what we're delivering to our you know, neurons, and these are essentially non-dividing cells. So one delivery event could basically place a small capsule of tRNA in that cell for a very, very long time. That would just make the reparative tRNA within the host cell. And that would essentially just serve to repair SCN2A as it's being made. Also, given that the possibility that the data that we have is currently showing, we're not seeing a lot of off-target effects. So, you know, we would hopefully have a one-time delivery would be what we're going for.
Great. And what other studies have you done using tRNA that has proven very successful to the point where you're moved closer to clinical trials? What other diseases?
Well, you know, to be honest, SCN2A is our main target. That's really the one that we're pushing after the hardest. We're working with other groups, and one of their main targets is a closely related SCN1A gene in Dravet syndrome. And so those two programs are pretty neck and neck in terms of how fast they're moving. The thing is about this technology is that there are, I think, an estimated 400 different genes that have PTCs neurologically. And so that's a large grouping of different types of neurological disorders, genetic neurological disorders. The delivery for any one of these, SCN2A, would most likely also be similar to the delivery for all the other ones. So you need not necessarily be focused on the other ones for them to benefit from research programs that are targeting PTCs. So SCN2A and 1A are clearly what we're going after right now, but if there's success in a clinical space, you can imagine very quickly how those different methods could be rapidly adapted to many other PTCs.
Great. So how did you decide on like studying SCN2A? Why SCN2A versus other channelopathies?
Oh, I've long been fascinated with sodium channels. For 25 years, that's been the main drive of my lab and of my research career, is to study and understand the function and physiology of voltage-gated ion channels. I was fascinated with them early on as a graduate student, realizing their potential to control electrical excitability throughout the body and excitable tissues of nerve and muscle. And it's been a passion of my life for a very long time. And so SCN2A falls very clearly in that area of what we've been excited to work on. So it's a real pleasure as an academician, as somebody who's running a research lab, to train physician scientists and graduate students, that we have an opportunity to merge a gene correction program which serendipitously arose from our basic research and apply it back to our core area of study, which are voltage-gated sodium channels.
Wonderful. Well, we're the lucky benefits that will, the recipients of your studies. So thank you so much for your passion and dedication. And how has our foundation impacted you with your work?
Well, it's been there's been on multiple levels. On one of them, very practically, just having access to some iPSCs and knowing the database. So that's a practical area, but it's also an inspiration to work with the SCN2A Foundation. It really is. I was at the meeting last year, and it was just so invigorating, and just really just brought everything into focus so clearly on why we work in the lab and why we stay late and why we do what we do, is to try and make a difference. And so the foundation itself really pulled together all those stories for us to see how important is the work that's being done. And so, it's practical and it's inspirational. So what you guys are doing is fantastic.
And with just asking you about the challenges versus benefits of working in an academic institution, and I know you mentioned the needing industry to speed up this process, but can you just tell us more about that, just in your setting?
Yeah, so the way that I see it, and I think everybody may see this relationship a little bit differently. But you know, academic settings is really where a lot of the basic ideas come from, and they get tested. This is where we try the crazy stuff. It's not always eureka that is the inspiration or the tip off that you have a new discovery. But oh, that's weird. Is when you say something, when you hear somebody in the lab say that, you know you may be onto something. And because we are given the bandwidth to really follow up on these one-off discoveries, we can build data sets, and then we can really take those to biotech and show them, you know, that these are solid and robust observations that are worth following up on. And then, you know, they have to secure the extra funding and take those discoveries across the finish line to the clinic. So it's really a partnership. There's no way an academic lab could do this on their own. But because so many of the biotechs that you see out there today, they're all working with technologies that came out of different academic labs. Very rarely do biotechs on their own make those types of discoveries, but they're able to pick them up when they see them. And so it's exciting to see that happen with this technology, that maybe it will be carried over to the clinic through that type of partnership.
Definitely. Now, we spoke about the call to action, which would be for families to mention to their, you know, providers to try to get the cells, you know, for research and whatnot. What else could you encourage our families to do?
Oh, I wouldn't know. I mean, I'm in awe of the families so much. I couldn't imagine you doing any more. Whenever I talk to SCN2A families, you just are so inspirational what you guys do. So, I mean, hang tough. Know that there are people out there that are trying to make a difference. You know, that it might not be us, it might not be somebody else, but there are people that are trying. And you know, just know that you're not alone. That what the gene issues of PTCs are with many people. So we call them rare diseases, but that's not really true. Taken together, there's a common cause there.
No, thank you for the words of encouragement and for your work and dedication again, and for your time. Thank you. This is amazing.
Thank you so much. Bye-bye.